Hi, this is Dr. Collins Jones. I created this series of videos on manual cell counting and cell density calculations in the hope it will be useful to my current and former students, as well as to anyone else who may want an introduction or a refresher for these basic cell culture calculations. I recognize there are many different approaches to explaining this material and to solving these types of problems. I am presenting the approach that seems to work the best for my students. Please use whatever approach works best for you. I have divided the entire manual cell counting and cell density calculation video series into a total of 7 segments. I will do my best to keep each segment 30 minutes or less. If you are taking the time to view these videos, I hope that you find them helpful. Alright, so segment 1, let's get started. First, let's do an overview of what I plan to cover in this series of 7 video segments. We want to go over the steps required to obtain a manual cell count. We want to define cell concentration, also known as cell density. We want to know what types of equipment and materials we need to count cells manually. We want to learn how to calculate the percent viability, how to calculate the cell density or cell concentration, how to calculate seeding concentrations for when we passage cells, and how to calculate batch seeding when we need to make the same concentration of cells in multiple flasks. So let's get started with an overview of manual cell counting. The goal here is to determine the concentration of cells in a growth vessel and the percent viability of those cells. Cell concentration, also known as cell density, can be expressed as cells per centimeter squared if we are dealing with anchorage-dependent cells where we have to worry about the surface area of the vessel that the cells are growing in. Or in the case of suspension cells where we're growing them in a shake flask or a bioreactor, we want to calculate the cells per mil. The percent viability distinguishes live cells from dead cells, right? So live cells look normal and healthy. Dead cells, we use a dye staining method. Trypan blue is really what I'm going to focus on here, but we have a viable dye. Dead cells take up the dye and turn blue. So the percent viability is then expressed as live cells divided by total cells times 100%. And again, we're going to illustrate these calculations with numbers and go from manual counting all the way through the calculation. So that will be in the upcoming segments. What's the equipment you need for manual cell counting? We need a hemocytometer. We need an inverted microscope. That's what we use. We use P200 and P20 pipettemen with the appropriate tips. We use a balanced salt solution, either phosphate buffered saline or Hank's balanced salt solution. So PBS, phosphate buffered saline, or HBSS, Hank's balanced salt solution. Some people dilute all the way with trypan blue. We do not recommend that because trypan blue sometimes is insoluble, and so you end up with crystals in your counting field, which can be mistaken as dead cells. Okay, so we do use, though, for this trypan blue, we use pyrogen-free microcentrifuge tubes to make sure we don't kill the cells that we've put in the tube. And in order to obtain a sample, we use 2 or 5 ml sterile serological pipettes. Let's look at the steps then involved in manual cell counting. Since we try to mimic a GLP or cGMP regulatory environment, the first step in any process for us is to record traceability following good documentation practices. Then we have our sample of cells. In this case, these are anchorage-dependent cells. They are attached to the flask. If it's a suspension culture, you just remove a well-mixed aliquot of the cells. And by that I mean, if you take the cells off the shake flask, then you want to remove a sample as quickly as possible because cells in suspension will settle and you'll get a gradient of cell concentrations from top to bottom if you do not work quickly. So here we have anchorage-dependent cells. We're going to need to detach the cells if they are attached. This can be done most often using trypsin, but occasionally people will scrape the cells. They will use a cell scraper. That can damage the cells, so it will decrease your percent viability. And scraping also often causes the cells to clump together, and clumps are difficult to count. So we recommend that you avoid scraping whenever possible, but some cells do not detach using trypan blue, and scraping may be your only option. Okay, so we've collected our cells. We're going to put these into our pyrogen-free microcentrifuge tube. If necessary, we're going to dilute the cell mixture using a balanced salt solution. So you can see they've sort of spread out here. Then we're going to add our trypan blue vital dye, and the dead cells whose membranes are presumably compromised will take up the dye, while the live healthy cells will exclude the dye. And so what you can see here is that after 2 minutes, non-viable cells will take up the dye. And if you read most of the manufacturer's directions for trypan blue staining, it does require a 2-minute timing in order for the dead cells to take up the dye. But if you wait too long, live cells will also begin to take up the dye. So the incubation time becomes important, and that should be part of your documentation practice. We're going to continue this then on the next slide. So we've got our cells, they're stained, they're diluted. We're going to load the hemocytometer. So if you look over on the hemocytometer, you'll see that I put some cells on there. There's some live cells, and then the blue cells are the dead cells. We count under the inverted microscope on medium power. We count 1 square at a time, counting both live and dead cells simultaneously. Ideally, we will count 10 squares, that is 5 squares per chamber, and in the next segment, we will define what we mean by squares and chamber. There should be no more than 50 cells and no less than 20 cells per square. If you are below 20 cells per square or above 50 cells per square, your count will most likely be inaccurate. Okay. Also, counting should be completed within 5 minutes of adding the trypan blue. Okay, so for manual cell counting, we use the cell counter here that I kind of drew a picture of. And again, we just want to emphasize that counting should be completed within 5 minutes of adding the trypan blue. After that, you risk healthy cells beginning to take up the dye. Then we're going to calculate the cell concentration and record that and report it as necessary. Then we want to clean the hemocytometer and return it to the case. This includes the cover slip. On hemocytometers, the cover slips are reusable. So please do not discard the cover slip. Clean it carefully along with the hemocytometer. And we clean our hemocytometers with a soft lint-free cloth, often a Technicloth, using 70% alcohol. Do not use abrasives, so do not use any solid detergents because they may scratch the hemocytometer, which will make it difficult to focus on the cells. Then we're going to turn the inverted microscope off, unplug it, and cover it. And of course, we always follow our 6SC principles in the lab. There are lots of semi-automated and automated cell counting instruments, right? And this is a partial list of vendors. Okay, I'm not going to read them to you. You could pause this and look at them. But I do want to emphasize this list is not an endorsement, and it is certainly not complete. There are more and more semi-automated and automated cell counters coming on the market every year. Many of them are excellent. These are the ones that showed up when I did a quick Google search for cell counters. Okay, and then the asterisk ones, the ones with the asterisks, are the ones that we have in our current biotech laboratories and use. And that is the end of segment 1.